Hello, welcome back to my channel, Bio with Shansi. In today's lesson, I want us to talk about cell signaling. This is chapter 4 of the Cambridge AS syllabus, that is cell membrane and transport. So, what is cell signaling? When someone talks about signaling, you can think of it as sending a signal. And signaling is simply about getting a message from one place to another. Remember, you're made up of millions of cells, and there is need for constant communication between those different cells so that there is coordination within your body. So with cell signaling, we look at how our cells use molecules to detect and respond to specific stimuli. The stimuli could either be external, that is, changes that are happening externally outside your body, or the stimuli could be internal, that is, changes happening within your body. So there is need for the cells to be able to communicate. But the question is, how do these cells really communicate? How do they send messages around the body? Why do we even need these cells to communicate? Remember that we are supposed to be able to respond to our environments. In case any change happens in your environment, you should be able to respond to that change. An example, you could become thirsty. When you become thirsty, it's simply an indicator that, you know what, you need to drink water, meaning your body has less water. So, how does your body respond to that change? It is important that we have signaling pathways so that they can coordinate activities of the cell such that responses are made in time. Remember, if your body doesn't respond to those changes in the environment, it can lead to your death. So we have different ways in which messages are sent around the body. We have the nervous system, which uses electrical impulses. And we also have the chemical or the endocrine system, which uses hormones. So in this case, we are going to look at how hormones or how those chemicals are used in sending messages around the cell or from one cell to another. So when it comes to the electrical way of sending messages, that one you'll learn about it in 8.2. But for now, we are going to concentrate on how messages are sent around the body using chemicals. And the process involves three main stages. Number one, we need a stimulus. A stimulus is a change in environment. I've given you an example of feeling thirsty. Feeling thirsty is an indicator that your body is having less water. So the stimulus is that the water potential of your blood has decreased. And when that happens, different parts of your body are going to be able to respond to that decrease in the water potential of blood. We can have another example of a stimulus. It could be maybe you have taken long without eating food, so the glucose levels in your body decrease. So the stimulus will be reduction in glucose levels. Now, different cells in your body will be able to detect the reduction in glucose levels. An example, we have the pancreas. It will detect the reduction of glucose in your body, and it's going to secrete a hormone. So that is what we are meaning by a stimulus causes cells to secrete a specific chemical called a ligand. An example, the stimulus is low glucose levels. What happens? The pancreas is going to be stimulated to secrete a hormone called glucagon. So in that case, glucagon is an example of a ligand. Ligand just means chemical. Yeah, it's a chemical compound. So we are going to have the pancreas secreting glucagon. So glucagon is going to be carried within blood to the liver. And that means that the liver is the one with cells that are ready to receive that glucagon. Once glucagon reaches the liver, it will attach to that liver. And then the moment the liver receives glucagon, it's an indicator that, hey, the glucose levels in this person's blood are really low. So the liver is going to be stimulated to carry out different reactions so that we have the glucose levels rise again. So when you look at the stages in sending of those messages, especially where chemicals are used or ligands are used, we need a stimulus. So the stimulus leads to secretion of a ligand. Then the ligand is going to be transported to cells that are needed to respond. And usually these target cells are very specific. An example, glucagon will mainly be sent to the liver, so the liver is the one to respond. Other parts of the body won't respond. Now, when the ligand reaches that target cell, what it does is that it binds to the receptors on the target cell. Let's use this diagram to further understand that. Yes, we've had a stimulus. An example, we've had a decrease in glucose levels in your blood. So a ligand is secreted. An example, it could be glucagon secreted by the pancreas. So the glucagon is going to be carried in blood. When glucagon reaches the liver, it has to enter into the liver. However, you'll find that sometimes the ligand is too large and it can't pass through the cell surface membrane. So we are going to have special receptors. And usually these receptors are spanning the whole surface membrane, like you can see this protein receptor. Now the shape of the receptor is always complementary to the shape of the ligand. That is why these ligands are very specific in nature, because it's because of shape. So they are received by specific cells, which we call target cells. So the ligand will bind to the receptor. The moment the ligand binds to the receptor, usually it leads to change in the shape of that receptor. When the receptor changes shape, sometimes it can activate the next component. Like an example here, we have a G protein. So when the receptor changes shape, it switches on this G protein or activates this G protein. Then the G protein also activates another protein, which is an enzyme that makes a second messenger molecule. Now, this enzyme is going to start releasing second messengers. Now, what are second messengers? Remember, we had a message coming in, which was in form of the chemical, the ligand, glucagon. But glucagon isn't able to enter into the cell surface membrane. But we need the message to reach inside the cell. So usually we have these small molecules called second messengers released. Because the first messenger was the glucagon. Now we can take these small molecules to act as second messengers because they are transmitting the message within the cell. So these second messengers usually are going to be released when they are many and they are small, so they can diffuse through the whole cell, sending or spreading the signal, spreading the message. Now, these second messengers can activate more enzymes. Then those more enzymes will activate other enzymes. Then those other enzymes might activate other enzymes up to when we have the last, we could say the last component of that cell signaling process activated so that we have a response made. Responses can vary. It could lead to maybe the cell carrying out secretion. It could be that maybe the cell needs to carry out transcription so that certain proteins are made. It could be that some movement is going to happen within a cell, or it could be a metabolic change. For example, in the liver, we might have a reduction in the rate of respiration so that we have less glucose used because of the example I was using, which was release of glucagon. So when you look at this cell signaling pathway, number one, we have seen that it can involve a ligand carrying the message. The ligand usually attaches to the receptor. What allows it to bind to the receptor is the fact that the shape is complementary to the shape of the receptor. The receptor will change shape and then activate the next component in the pathway, which is usually a G protein. Then the G protein will activate an enzyme that makes second messenger molecules. So that enzyme will lead to release of very many second messenger molecules. Now, at each stage of this signaling pathway, we see what we call amplification. Amplification means increasing something, amplification. Like an example, you could be talking and I tell you amplify your voice, meaning increase, speak louder. Now, we had just one ligand binding to the protein receptor. However, that led to release of many second messenger molecules. Then the many second messenger molecules lead to activation of many enzymes. The many enzymes will activate other many enzymes. So we call that amplification. So at each level, the message becomes bigger and bigger and bigger. The message is amplified. We had one ligand, but many second messenger molecules were released. Then many second messenger molecules activated many enzymes activated many other enzymes. So that is what we call amplification. Now we have another characteristic still of this pathway, which we call a cascade effect. We have a cascade. A cascade is like a chain of reactions. A chain of reactions where we have one thing leading to another, one thing leading to another. An example, we saw the receptor binds with a ligand, then that leads to change in the shape of the receptor activates a G protein, the G protein activates a second messenger releasing enzyme, then the enzyme releases second messengers which activate other enzymes. So it's a chain. One reaction leads to another. So we are having a signaling cascade. So this type of signaling that involves a ligand usually has amplification at each level, and then it is also a cascade, a signaling cascade, because we have one thing leading to another. We have a chain of reactions happening, and that is how messages are sent inside a cell. So someone could be wondering, why is it that these ligands cannot enter directly into the cell? Some of the ligands could be polar. If they are polar, they cannot pass through this hydrophobic core because they will be repelled by the hydrophobic tails. So ions, polar molecules like glucose, they all large, if sometimes even the ligand could be large, too big that it cannot pass through the cell surface membrane. Still, in that case, it would just need a receptor to bind to. So basically, that is what we've been looking at. We have seen that the cell surface receptor has a specific shape, which is complementary to the shape of the ligand. And it is only cells with those receptors that will be able to recognize the ligand because of shape. When the ligand binds to the receptor, it brings about change in the shape of the receptor. When the receptor changes shape, it interacts with the next component, and that is how the message is transmitted into the cell. Yeah, that is how the message is transmitted into the cell. And we have also looked at the G protein. Once the G protein is activated, it switches on the enzyme, which releases a second messenger molecule. Once we have the second messenger molecules released, second messenger molecules also activate more enzymes. And we have amplification. I've already talked about what amplification means, which still means magnification. The message becomes bigger and bigger because many more other molecules are activated within the cell. And we have also seen that we are going to have a signaling cascade, a chain of reactions happening as the process of signaling is occurring. So usually cells respond to signals or cells respond to ligands by release of a second messenger. We have seen that signaling pathway. Other times, we might not have a second messenger released. We can also have maybe other processes activated within a cell once the ligand binds to the receptor. So sometimes when the ligand binds to the receptor, we could have an ion channel opening within the cell surface membrane. Remember, we shall look at channel proteins. Yeah, we shall look at channel proteins, but sometimes we can have a channel opening within the cell surface membrane, assuming this is my cell surface membrane. So the moment the ligand binds, assuming this is the region where I'm having the protein, so I have binding of the ligand, we could have a channel protein open so that we can have maybe ions entering into the cell. Maybe they could be sodium ions, and then a reaction can occur or the needed response can happen. We could have some of those receptors acting as enzymes catalyzing reactions which lead to the appropriate response being made. We could also have some of the receptors. Some receptors could be inside a cell. So not all receptors are just found on the cell surface membrane. Like an example, let's use this picture to understand what I am talking about. So this picture still shows us a cell. They're showing us that when the ligand attaches to the cell surface membrane, what happens? We can have the cell responding in different ways. Sometimes we have seen we could have a second messenger molecule released, or some ligands are able to cross the cell surface membrane and enter inside the cell. Like in this case, such ligands are said to be hydrophobic. So if they are hydrophobic, for them, they can be able to pass through the hydrophobic core of the cell surface membrane without being repelled. So such ligands will be able to enter directly into the cell, and that means that their receptors are going to be found inside a cell. And then once they bind to their receptors inside a cell, then we can have a response made. Different responses can be made. Sometimes the ligand could lead or activate the cell to carry out secretion, release certain substances. Like here, the substances were able to pass through the cell surface membrane directly. That means still that these substances being secreted are hydrophobic in nature. But then in other cases, we could have some of the substances in vesicles, and then they are later released outside the cell by exocytosis. So what you need to know is that molecules that are hydrophobic usually can cross the cell surface membrane without being repelled by the hydrophobic core. So cell signaling can also involve what we call direct cell-to-cell contact. What is direct cell-to-cell contact? So here we are going to need cells to come in contact so that a response is made. Like an example, you learn about this in immunity. When you look at lymphocytes are cells that are able to defend our bodies against pathogens. So sometimes these lymphocytes are going to make a response after they have come in contact with a pathogen. So the lymphocytes have receptors on their cell surface membranes whose shapes are complementary to the antigens on the pathogen. So the moment the receptors of the lymphocytes come in contact with the antigens on the surface of the pathogens, then the lymphocytes are going to make the right response. They will either divide and increase in number and then start secreting antibodies that are going to be used in fighting against those pathogens. So basically, that is how cell signaling occurs. If this video was useful to you, please give it a thumbs up and then feel free to share the content with your other friends. Thank you for your time. See you again on the next one.